So yeah, just if you're joining us right now, we have Paula Morais and Pedro Farias from the Universidade de Coimbra. And you're also at the Centro Ciencia Viva de Coimbra and part of the Exploratório team I have written down here. And you're going to be giving us a talk on pet microbes.
Pet microbes, yes.
Yeah, okay. Do you need to share your screen for any presentation, or are you able just to give it in person?
I will share the screen, and I think that Pedro can read from my screen, yes?
Yes,
yep,
yes,
you would be able to see it.
We'll take turns.
Yeah, okay.
Take a second, and I'll join you in.
Okay, I will start, okay?
Fun, yeah. So you have until 8:20. So I'm going to turn off my video to avoid distracting anyone. And once we have Pedro return, you may begin. But yeah, you're the first live talk in this kind of daytime session now. We've gone through the night, and now we're, at least in Europe, in the daytime. So it's all quite exciting. Okay, are you both ready?
Yes.
Both ready.
Yeah, okay,
yes,
great. Well, begin with pet microbes. We look forward to your talk.
Okay, thank you. Sorry, but I cannot share.
Okay, let me just check. How about now?
Because
are you able to share? If you try the share screen button, it
okay,
should work.
it's now. I can share it now. Thank you.
Cool. Ah, perfect. We can see. Great.
Okay.
Let me. Sorry, because I cannot. Have here some problems with. Okay.
No problem. These are all part and parcel of a live talk.
Yes. Sorry, because I think I opened the wrong presentation. It will be easier like that. Okay. 1 minute more and a few seconds, and that's it.
Fantastic. Right.
Okay. Thank you.
Here we are. All looks good.
Okay, so good morning, everybody. We are talking about pet microbes, and at the end of the presentation, we will see why we are talking about pet microbes. I want to start by talking a little bit about our university. It's not really a technical presentation at this time of the day. It will be difficult. So it's about our university and our famous microbiologists and our famous microbes at the University of Coimbra. The University of Coimbra, it was created in 1290 by one of the oldest king in Portugal, and it is one of the oldest universities in Europe. It was was stabilized in Coimbra in the 16th century, and nowadays our university has about 25,000 students. It's old, but it's not stopped in the time. So today we have, as I told, around 25,000 students, and it's dedicated to research. It has a lot of research projects, national and international projects. And it's also an international university, since it has a lot of foreign students. Many of them, I don't want to say most, many of them coming from Brazil and other Portuguese-speaking countries. Although we belong to the Erasmus group, and we have also many students from many nationalities. In the University of Coimbra, we have the first culture collection dedicated to bacteria in Portugal. It is registered at the World Federation Culture Collections, and for us as microbiologists is an important asset to work on microbiology. Our group is the group who runs the culture collection, and because it's a research group, it's really makes the collection more active and enables the collection to be an integrated system ranging from the isolation to the classification of new organisms. It is also a training system, so we have many students related to the culture collection and enable the culture collection to be also a way of training students. Our culture collection has more than 2,000 strains that have been isolated by different groups of microbiology in the university, and those strains are being characterized and described as new species or not. We have a tradition of describing species, as you will see, and these strains give us work on microbiology and enable us to contact with companies and industry to make these organisms potential biotechnology tools. We have also at the University of Coimbra famous microbiologists, and I want to talk a little bit about Milton Cochella. Milton was president of the Federation, the European Federation of Microbiologists Society from FEMS, and he was a real traditional in the way that he worked on taxonomy and on microbiology and on description of species before the genomic area. So he was really dedicated to his bugs. Going now to the famous microorganisms in our lab. During the last years, we have, as group of environmental microbiology, described new species as a tradition of our university that starts with Milton. And we want to talk to you about one famous microorganism in our lab, which is the species we described, the species Glacimonas singularis. This strain was isolated from a uranium mine wastewater treatment plant. We work as an environmental group in many metal contaminated environments, and we isolated this strain from the wastewater mine treatment. And the strain was characterized by resistance to uranium and by the ability to be in relative high numbers in the system. And I now ask Pedro to talk about our famous Nosocomicoccus ampulle.
Thank you. Late in 2013, this new strain, Nosocomicoccus ampulle, was identified, was isolated from in a work with a collaboration, and it was isolated from a sample of a surface of a bottle of saline solution from an hospital. So there were two strains, and these two strains isolated were later characterized and identified as a new branch on the Bacilliaceae family. But the work on characterizing new species and genus didn't stop, and then we moved on. And in a more recent study, move to the next. In a more recent study, a colleague of ours from the lab, and in collaboration with Professor Paula and some other researchers, isolated microorganisms from a project that was characterizing the microbial endophytic community of Pinus penasters that were at the time affected by the pine wilt disease, and a strain was identified and characterized, and this strain was later on observed to be belonging to a new family, and this was a great discovery for the group. And contrarily to the previous identified strains that we present here, this one already had the contribution of the new techniques that the laboratory employs, like genome sequencing and some other new and modern techniques for characterization. And so we can move on now to the exhibit that we are preparing. And I don't know if Professor Paula wants to take it, the beginning.
The exhibition we are preparing. It will be present. It will be available today. Please, Pedro, help me.
I think it starts today at 10 or 11, the opening. So it will run through some days or a couple of weeks. I'm not really sure.
I think we don't know. We are not really sure because it is a live. It is a live. It is a live exhibition in the real way, in the proper way, which means that the organisms are alive. So
they are presented in petri dishes, live grown cultures, and these are just few examples that we have in exhibition down at the Exploratorium that you see in here in the image.
So together with the microbiologists of the other faculties of the University of Coimbra, we belong to the Faculty of Science, and also with the Veterinary School of Coimbra and the Center of Life Science of Coimbra, which is called Exploratório, we organized an exhibition of microorganisms. So you can go to this exhibition and look at live cultures. You can go more than once, so culture will be different during the exhibition. It will be there at least 1 week, so people can go during this next week to the exhibition. And we select the organisms that are present at the exhibition with the criteria which of the organisms it's more related to me. So I select 2 organisms, Pedro select other organisms, and all our colleagues select at least 1 organism. We also ask to the students to select 1 organism, so we can go to the exhibition looking to our organism. So for me and Pedro, we decide to talk a little bit about Pedro, Bacillus altitudinis.
So real short because we are running out of time, but one of my pet organisms is Bacillus altitudinis, and is my pet organisms because it's one of the organisms, one of the bacteria that has been following me in my PhD. And this is a very interesting organism, it's Bacillus. It was isolated from a mining sediment in a deactivated mine in Portugal, and it has a very high level of resistance to heavy metals. One of them in particular is tellurium, which is the focus of my PhD dissertation. And this bacteria has a high capability of reducing tellurium and in the process fabricating very structured tellurium-containing nanoparticles. So in the exhibition, you will be seeing me detailing this bacteria, looking through this and plate where we have the bacteria, with a regular phenotype moving to a phenotype where it has tellurium in it, and you'll have a good look on site of this bacteria, my pet bacteria. So, Professor Paula?
Okay, my pet bacteria, the one I select is the strain called B2A2W2, which is already a robot before being it. And it's from the genus Diaphorobacter, and from the species, and this is the difficult name, Polyhydroxybutyrativorans, which is a very, very long name, and actually it describes a little bit the strain. It is able to degrade polybeta-hydroxybutyrate. So this is quite known. It's known only since 2015. And this strain was isolated from sediments, from tailings of one mine in Portugal called Panasqueira mine, which is in North Portugal. And the strain, it was isolated during our project Biocritical Metals. And the aim of the project is to study the ability of microorganisms to extract metals from mine waste, and to evaluate their ability to bioleaching, which is the word for metal solubilization, and this way evaluate the possibility to use mine wastes as secondary sources of metals, contributing to a circular strategy for raw materials. Now, our strain Diaphorobacter is able to solubilize iron, zinc, and copper, and in high quantities, and is really, really a nice strain. You can see in this picture below here, Pedro, catching samples from the mine, and we hope that after a long study about the genetics of this organism, we will be able to transform this microorganism in biomicrotechnology. So to end the talk, I want to thank you, all my students and all the researchers that belong to the group Environmental Microbiology. And I want to thank Pedro for this really, really morning. And Exploratório that allowed us to exhibit our microorganisms. Thank you very much.
Thank you.
Thank you very much. I actually have some pet microbes with me today. So I have
you already have
a selection of alga and I think some cyanobacteria and lots of rotifers. So we'll see these later. But just so everyone knows, so the Exploratório is open today. So you could go and see the pet microbes if you're in the area across all of today while it's open. Fantastic. So I can see my next guest is in the waiting room. So I need to move on to the next section. But thank you so much for this wonderful talk and getting us off to a fantastic start.
Thank you very much.
Of the daytime part of this live stream. We've already been going for about 7 hours, but we've got plenty more to come. So thank you very much. Enjoy the rest of International Microorganism Day. And hopefully some people can see some pet microbes today.
Yes.
Thank you very much. Bye-bye.
No problem. Bye.
Bye.